Welcome back, biologists. So, today we're going to talk about how we make our cells, how we prepare them to stain them, and how to see them in a microscope. So, a lot of this is stuff that you actually do in lab. So, if you weren't able to attend lab and do these in person, please make sure you're paying careful attention. And if you are doing this in lab, please pay attention so you're doing the correct technique and protecting yourself. So, when we're talking about making a smear, we're talking about putting a thin layer of microbes on a slide. We only want a thin layer so that we can see one without having other ones piled on top of it. And one thing we're going to do is fix them. Fixing them means we make those microbes stick to the slide. We don't want them to just rinse off and go down the drain and have nothing left on your slide. Fixing also does the nice thing of killing the microbes, so you're not contaminating yourself or the microscope or anything when you're using that slide. There are two ways to fix a slide. One is heat and one is alcohol. Heat is how we fix it in our lab. Although we'll take this slide here and stick it onto a clothespin and pass it through this flame a couple of times. Using a clothespin lets your finger be a little bit further away, so you're less likely to burn yourself. Then once it's been smeared and fixed, you can stain it. When you stain it, you are coloring that microbe. You have some sort of dye that will make it show up, because an unstained cell is mostly colorless and just blends right in with the medium. There's two different kinds of dyes, basic dyes and acidic dyes. Basic dyes have a color on a positive ion, which is really good to stick to bacteria because bacteria have a little bit negative charge. The one we use most in lab is crystal violet. So crystal violet has a slight positive charge, sticks to that negative charge in the bacteria. Acidic dyes are often used for negative staining. Negative staining is when you use a color, when you color the background and leave the bacteria unstained. So by doing that, by not staining the bacteria, you can see how living cells behave without having some sort of dye stuck in them or killing them. A simple stain is going to be a solution of just one single basic dye. It's going to color everything the same color. So it's basically there to see what shape the microbes are, how the different microbes attach to each other, and you'll be able to see basic structures of it. Sometimes with a simple stain, we add a mordant. A mordant is something that intensifies the stain. Mordants could do something like increase the affinity of the stain for a specimen, so make it stick to the cell a little bit better. It might do something like coat a structure, so it's part of the cell that's thinner or smaller. By coating it, it's easier to see it. Iodine is a mordant that we'll be using in gram staining. So iodine helps that crystal violet stick to gram-positive cells. And then a trapping agent might help stop the dye from being removed. That's how iodine works, is it stops the crystal violet from being removed. Instead of just simple staining, where you stain everything the same color, we can use differential staining, where things will look different. They use different types of stains. So differential staining lets you tell different things about bacteria. One type of differential staining is the acid-fast stain, which lets you know if a cell has different types of waxy molecules in their cell wall. One type of differential stain is a gram stain. A gram stain lets you know if your cells have a thick cell wall or a thin cell wall. And the reason you care is that gram-positive bacteria, which have a thicker cell wall, tend to be killed by penicillins and detergents. Gram-negative bacteria with their thinner cell wall and an outer membrane are more resistant to antibiotics. So if you don't know what kind of bacteria you have, you don't know you don't know if you can give your patient penicillin or not. So that's a big reason why we care about the gram stain, because it makes a difference in how you can treat that infection. Gram stain is very important for you to know. This is something we'll talk a lot about in lab. You'll have a lot of time to do it. We talk about it in chapter 3 and in chapter 4. If you have mastered this content, you will be able to recreate this this slide from memory. So you should be able to tell me the different steps of the stain: crystal violet, iodine, alcohol, safranin. You'll be able to tell me the purpose of each stain. So crystal violet is used as a primary stain. That's the first stain. Iodine is used as a mordant to intensify and trap the stain. Alcohol is used as a decolorizer, and safranin is used as a counterstain. So gram-positive cells and gram-negative cells, both, when you add crystal violet, will stain purple. Then the mordant iodine will not change the color of anything, so everything will still be purple. However, it'll make nice big crystals that will stick to those positive cells and won't rinse out. Then your alcohol is your decolorizer, and it will wash out that crystal violet. So at this point, your gram-negative cells are colorless. However, that crystal violet was trapped in your gram-positive cells, so they're still going to be purple. So at this point, you have purple gram-positive cells and colorless gram-negative cells. You can't see a colorless cell, so we need to add some sort of stain to it. So the last step of a gram stain is safranin, which will stain these colorless cells because it'll add red. That red stain of the safranin will not overpower the crystal violet, though, so those crystal violet will still appear purple. So here, can you find a gram-positive cell in this slide? Can you find a gram-negative cell in this slide? So the ones that retain that crystal violet are going to have this bluish-purple look. They're gram-positive. And the ones that lost the crystal violet that will appear pink or red from the safranin are our gram-negative ones. So here, all these little kind of curly Vibrio shape are our gram-negatives. An acid-fast stain is another kind of differential stain. The acid-fast stain will bind to bacteria that have wax, that have this waxy material in their cell walls. So acid-fast microbes will appear red. Acid-fast bacteria include mycobacteria and nocardia. Mycobacteria includes M. tuberculosis, which causes tuberculosis, and M. leprae, which causes leprosy or Hansen's disease. So if you suspect your patient has tuberculosis, you could do an acid-fast stain. Or if you suspect your patient has leprosy, you could do an acid-fast stain, and they'll appear red. So here, these are the little bacteria that cause leprosy showing up here. Then there are also special stains. Special stains are used to show up a particular special part of the cell. So an endospore. Endospores we'll come back and talk about in a little bit in chapter 4. Endospores are resistant dormant structures. Some cells form endospores when the conditions are not good to live. And instead of dying, they kind of hibernate in this resistant structure called an endospore. And they'll show up as these green spots. So all these green spots are endospores here, where the red is the growing bacteria. Then capsules. Capsules are a sugary coat around a bacteria that they're not going to hold on to a lot of dyes, and so they won't show up as any color. So you stain your cells, then you do a negative staining to stain your background, and your capsules show up as these little halos around it. So that's a capsule staining or negative staining. And then flagella. Flagella are the little arms, hair, string things that allow bacteria to move. They're structures of locomotion, but they're too thin to be seen just with a regular stain. So we're going to thicken them with a mordant, and then you can have the flagella show up. So that's a flagella stain. Okay, so then we've discussed simple staining, gram staining, acid-fast staining, negative staining or capsule staining, endospore staining, and flagella staining. Please be able to describe all of these to me. You can read this off of your slide or off of your PowerPoint. Be able to tell me what each of these stains is used for. Especially that gram stain. Know every detail of everything we have said about a gram stain. None of that is irrelevant. Every detail of that gram stain is important to know. Okay, so let's review some of the questions from this lecture part. Why doesn't a negative stain color a cell? Why is fixing necessary for most staining procedures? Why is a gram stain so useful? Which stain will be used to identify microbes in the genera Mycobacteria and Nocardia? And how do stained endospores appear in an endospore stain? Okay, as always, if you have any questions, please let me know. We are getting closer and closer to your first exam, and I would really love you to be well prepared to make a great grade. Okay, meet me next time when we start talking about what actually is going on in a cell when we discuss chapter 4. See you later. Bye.